Okay, can I get started, Katie?
Yes, please.
Okay. Hi, everyone. This is Shigeto Shieda, Professor in School of Natural Resources, and I'm hosting this lunch and learn seminar today. Uh, Before we get started, please let me make a brief announcement. One Health Initiative is planning global One Health Symposium on April 10th this year. Uh, we have already 200 participants from 20 countries, and the number is still going up. There'll be many presentations and breakout sessions on important One Health issues such as antimicrobial resistance, zoonotic diseases, and environmental toxins. So I will send you a link in a chat. So please visit the website and look at the content of the symposium, and please register if you are interested. So it's my pleasure to introduce today's speaker, Dr. Alaa Sewid. Dr. Sewid is a postdoctoral research associate in the School of Natural Resources with strong background in molecular microbiology and infectious disease research. She earned her DBM at Zagatec University in Egypt, followed by MS in microbiology at the same university, and later completed PhD in biomedical science and molecular biology through a joint program between UT and Zagatec University. Prior to her current position, Dr. Sewid served as a lecturer of microbiology at Zagatec University. Her current work focuses on the development of new molecular biological methods for pathogens that are important in One Health, agriculture, and food security. She has 90 publications with 2 recent publications on the pathogen detection methods and received awards from UT and American Society for Microbiology, and recently submitted grant applications as co-PI to USDA. With that, I'll turn it over to Dr. Sewid.
Thank you, Dr. Ida, for this kind introduction, and thank you all for joining us today. And I will be so happy to discuss my research about how we can rapidly detect infectious threat and antimicrobial resistance. So as you see right now, time is ticking. So infectious disease should be diagnosed in timely manner, and it can be enabling the choosing of appropriate treatment and clinical decision. Why from lab to field matter? Infectious threat and antimicrobial resistant demand rapid, doable, and actionable diagnostic outside centralized lab. So when we're talking about lab to field, we should differentiate between centralized laboratory and resource-limited situation. For centralized lab, long turnover time required and also limited testing capacity is happening. And for resource-limiting setting, there is a lack of complex equipment and limited availability of trained personnel. And this will leading to delayed diagnostic. Delayed diagnostic is very crucial, especially when the time is difference between life and death. The cause of delayed diagnostic is inadequate resource in resource-limiting setting and long turnover time required in centralized laboratory. This will lead to bad consequences, especially due to reduced treatment effectiveness and increased antimicrobial resistance. And this will leading to increased complication with longer recovery. And it may be lead to high mortality, especially in life-threatening conditions. And this will leading to financial burden on healthcare, especially due to prolonged treatment and requirement of additional diagnostic tests and recurrent hospitalization. Next generation molecular tool must move beyond sensitivity alone and deliver speed, simplicity, and decision-ready data at the point of need. So we should talking about the point of care diagnostic as a technology. Usually, The most of diagnostic tools are, are requiring to presence in centralized laboratory, and this leading to the testing and sampling site will be separate. But when we moving to point of care technology, it's aiming to moving the powerful diagnostic tool from centralized laboratory to be in the sampling site, to making nucleic acid detection more portable and easy to use. This will be allowing rapid and accurate results. There is a several critical bottleneck in diagnostic workflow that leading to hinder the transition of diagnostic from centralized laboratory to be true field-diable point-of-care platform. First of all, the requirement of an extended incubation period and isolation of microorganisms and culture enrichment that is incompatible with rapid decision-making in field setting. For example, culture enrichment will taking more than 24 hours to isolate the bacteria. Second, nucleic acid extraction will leading to multiple steps and incubation period and also complexity of commercial kit. This will lead to needing of trained personnel, and this is not compatible with field setting. Also, using of a nucleic acid amplification to using a gold standard PCR method is requiring a complex thermocycling, and also it is leading to extended processing time. Finally, the detection of amplified product is needing to having laboratory-based readout system that leading to further limitation for on-site deployment. This is why the conventional laboratory workflow must transform it to more simplified and minimal instrumentation using to become more suitable for true field-diable situation. And this is we are going to explain in our technology. First of all, we're using immunomagnetic separation to enable selective concentration of target pathogen from complex matrices that reducing the time requirement for culture enrichment and maintain specificity. Also, we're using heat lysis as alternative for complex commercial kit extraction for DNA extraction that is requiring minimal equipment and also reduces the time prolonged incubation period. And we depending on loop-mediated isothermal amplification technology as one of the most important isothermal amplification technology that not requiring thermocycling and reducing the complexity of an instrument. This developed in 2000 and composed of more than 43% for other technology using isothermal amplification. Loop-mediated isothermal amplification can be differentiated between PCR as it's required only constant temperature, while PCR requiring thermocycling, and also it's reducing the time for amplification for more than 1/3 compared with PCR, and it is required 4 to 6 primer required for target amplification comparing to 2 primer required for PCR. And for detection of LAMP endpoint results, it's requiring for gel electrophoresis or turbidity detection by turbidometer, or fluorescent detection, or pH-based calorimetric detection. And also for phenol red-based calorimetric LAMP assay, there is a key limitation that indirectly report the amplification through pH fluctuation rather than target-specific signal. This is leading to non-specific amplification and false positive results, and this is reducing the performance, especially in biological matrices. So we are going to using DNA enzyme as next generation detection strategy to able to using simplified instrumentation and also a visual readout that capable to compatible with electrochemical detection and smartphone analysis. So we're going to discuss the DNA enzyme structure. It is short single-stranded DNA sequence that exhibit peroxidase-like activity upon its folding to G quadruplex. DNA enzyme amplification can be done co-amplified with target without compromising the specificity and generating multiple copies. And the DNA enzyme detection It can be forming complex with a cofactor of amine to oxidize substrate, either TMP or ABTS, to producing visual stable signal that is only produced upon correct target amplification that leading to reduced false positive results. So our workflow are going to use all-in-one on-site detection system that able to making pathogen enrichment and isolation by immunomagnetic bead separation and enrichment, and also beam extraction and amplification by using portable heat block and producing amplified target that able to produce a visual stable color by signal on approach that can be analyzed by smartphone analysis with spot Excel apps on the smartphone or potentiostat for electrochemical detection. We are having established collaboration with a well-known industrial partner to have the all-in-one on-site detection system on lab-on-chip signal generation by integration of microfluidic device. And this can be compatible with electrochemical and colorimetric detection for more scaling in the industrial use. So going to detect different infectious disease as a diagnosis. So there is a different infectious agent, either fungus, virus, or bacteria. So first of all, we're going to discuss our method to detecting bacterial pathogen. For example, Shiga toxin Escherichia E. coli, or can be abbreviated as ESTIC. It is one of the most important leading cause for foodborne illnesses, and it is concerning the food safety. The ESTIC is carried asymptomatically in agriculture or food producing animal, and it can be shedding in the fecal matter, causing contamination of food and water and causing foodborne illnesses for human, and also can be transmitted from human to human. And in Tennessee only, there is more than 2,000 cases happen annually from ESTIC foodborne illnesses. This underscores the need of rapid on-site detection strategy. And there is severe outcome from ESTIC infection in human, including diarrhea and bloody diarrhea, and can be leading to hemorrhagic uremic syndrome in severe cases. So we are collaborating with the Michigan State University ESTIC Center and having several ESTIC strains, including the main serotype 0157 and the big six causing foodborne illnesses to test and validate our assay to detect the amplification of STX1 gene and compare the DNA enzyme LAMP technology with the gold standard qPCR. And we able to detect and validate our assay by producing a stable visual detection by signal on approach, and it's having a very comparable result with qPCR. And this is leading to high sensitivity detection in complex food matrices, either in lotus or milk. Using immunomagnetic separation, it's leading to rapid turnover time, less than 2 hours from sample to result. And this having excellent agreement with gold standard method, with good correlation between PCR and also kappa factor. And this having outstanding diagnostic performance of sensitivity and specificity and also accuracy. Moving for another bacterial pathogen that we are dealing with, Leptospira as a life-threatening zoonotic pathogen that impacting animal and public health. For something known about Leptospira, it is most difficult bacteria to be grown, so it is needing to having a very sensitive and accurate diagnostic tool. For that, we can talking about Leptospira, how they affect the public health. It can be carried asymptomatically in rodent, and this can be discharged in water bodies and affecting agricultural animal and also pet animal. And this can be transmitted to human and causes leptospirosis or Vidal disease in human. We're able to detect this pathogen by using Leptospira interrogans as our positive culture and compare this with another cross-reactive species. And this can leading to a high sensitivity and detection of Leptospira for 10^3 copies per reaction. And by using a spiked urine sample from canine source, we're able to detect the Leptospira by using heat slices comparing with commercial kits within rapid turnover time, less than 1 hour from sample to result. And moving for another bacterial pathogen as mastitis causing microorganism, for gram-positive or gram-negative mastitis causing microorganism. This can causing mastitis in animal, and this mastitic milk can infecting the human and causing foodborne illnesses. Besides, it can be discharged in fecal matter and causing environmental contamination, and it can contaminate food and water and transmit to human as well. So by using 3' set, we're able to detect staphylococcal species and streptococcus species as major gram-positive causing mastitis microorganism, and also the major gram-negative causing mastitis microorganism by stable color detection. And moving for another fungal pathogen, we're able to having an NSAP award with multi-institutional collaboration to have a hyperspectral imaging and AI-driven analysis for rapid detection of Cercospora zeamediasis as a fungal pathogen that affecting cornfields. And moving for targeting of viral pathogen of influenza type A viruses as an example for this target pathogen. So influenza type A viruses are negative sense single-stranded RNA virus with 8 segmented genome. And it has 2 surface protein, hemagglutinin and neuraminidase, responsible for continuous reassortment and interspecies transmission. This leading to more than $1.4 billion costing in poultry industry as a losses in 2025. And avian influenza are naturally a reservoir in wildlife aquatic bird that can maintain diverse strain and also can transmit this for agriculture, poultry flock, swine industry, and recently for cattle as well. And due to the carrier barrier ability of influenza virus and genetic reassortment and high mutation rate, it leading to development of high pathogenic avian influenza that can be genetically spill over for human and leading to flu illnesses in human. For CDC estimated 2025 result, the flu illnesses in human causing more than 80 million cases with more than 1 million cases for hospitalization requirement and leading to 100,000 cases for flu disease. And to compare the conventional current method for lab detection for high pathogenic avian influenza, there is a limitation, especially due to having single-stranded RNA viruses that require 2-step incubation for lab, requiring reverse transcription. Also, genetic reassortment and high mutation rate of influenza virus requiring complex primer design. For 6 primer, it will be very difficult for doing this for single-stranded with high mutation rate. That's why we try to eliminate the reverse transcription process and simplify the primer design to enable easy visual readout. So we are trying to following technology that's called extension-mediated self-folding technology that eliminates reverse transcription process by using only 1 step and also simplify the primer design by only using 2 self-folding primer. And by using different linker DNA enzyme in the self-folding primer, we're able to generate a significant signal-to-noise ratio and the visual readout by using new DNA enzyme called MSBDX3. And we can compare this by conventional non-normal exit and the DNA enzyme exit to producing color detection with shifting in the amplicon. And we're using also positive sense single-stranded RNA to test the specificity of our primer design. And this leading to detection limit as low as 100 femtomolar. So after we apply our detection method for pathogen detection for virus or bacteria or also fungi, we are trying to moving beyond this to guide appropriate therapeutic decision that it will be very essential in effective treatment and limit antimicrobial resistance. So how antimicrobial resistance are developed? So usually there is a sensitive strain and antibiotic resistant strain. When the antibiotic are administered, it kills the bacteria that are sensitive and leading to more space for growing of the resistant bacteria that can be transfer the gene of resistance later on for sensitive bacteria. How is this antimicrobial resistance are transferred for different public? For example, at hospital, patients attending hospital that having antimicrobial resistant strain able to transfer to another person. And also the prophylactic treatment with antibiotic or haphazard administration of antibiotic for food producing animal or food as a sample, it leading to propagate of more and more antimicrobial resistant strain that later on transfer by food to human. And this leading to more than 2 million cases of antimicrobial resistant infectious agent in United States per year. And this leading to more than 30,000 people die as a result. The conventional method for antimicrobial susceptibility testing is either phenotypic method to detect how the organism are respond to the antibiotic, either by dilution method or disk diffusion method, or molecular-based method to detect the presence of the gene resistance. And both of them are requiring more than 3 days to develop the result of antimicrobial susceptibility testing, especially due to needing for culture positive results and isolate the colony and then perform the antibiotic susceptibility testing. And also, is the antimicrobial resistant diagnostic should be reported the function resistant rapidly and not only required to detect the gene of the presence. That's why we try to follow a viability-based molecular assay that is depending on using photosensitive dye that able to penetrate the non-viable cell and not able to penetrate the intact cell membrane of the viable cell. And it's able to covalently bound to the DNA for non-viable cell and prevent this from amplification. This leading to differentiation between viable and non-viable cell by PCR method. And we try to adapt the same method to differentiate between antibacterial susceptibility bacteria and antibacterial resistant bacteria. So antibacterial susceptible bacteria are usually affected by the antibiotic, leading to growth inhibition or compromised cell membrane, while the resistant bacteria having intact cell membrane. This leading to avoid the photosensitive dye or BMX to integrate into the cell membrane and leading to successful amplification of DNA that from resistant bacteria leading to signal on approach, while the susceptible bacteria having immunocompromised membrane or affected membrane that enable the integration of photosensitive dye and covalently bound to the DNA and leading to inhibition of DNA amplification that leading to signal of approach. So we can simply differentiate between antibiotic susceptible and antibiotic resistant strain by using signal on and off approach. So we're able to apply this method to differentiate between different clinical E. coli isolates and comparison with the sensitive reference strain and detected the antibacterial susceptibility testing for two antibiotic, ampicillin and tetracycline. So any any strains that are producing signal on approach, this means they are resistant for this antibiotic. And this can be using a very clear demarcation between the susceptible and resistant with good signal to noise ratio. And this leading to a very good sensitivity for combined detection of antimicrobial resistant for ampicillin and tetracycline for more than 91% for sensitivity and close to 85% for specificity and accuracy. So one molecular diagnostic platform can be using minimal instrument and can detect different target, either bacteria, virus, or fungus, and also can be profiling the antimicrobial resistant. The implication of our study is to lab to field transition impact. First of all, rapid diagnostic can shorten the detection time from days to hours, and also provide simple and robust platform that maintains the accuracy to the point of the need. and expanded the access to diagnostic where the real-time decisions are made, not only the infrastructure exists. For clinical and public health impact, early identification of a pathogen and the antimicrobial resistant marker is leading to guide target therapy and also real-time surveillance and antimicrobial resistant monitoring during outbreak of emerging and re-emerging pathogens. Improved clinical decision making and reduced inappropriate antibiotic use, thus strengthening the antibiotic steward. So its impact on One Health and global health, integrated diagnostic across human, animal, food, and environmental health sector, enhance cross-sector surveillance, enabling good data collection and more equitable global access for advanced technology for diagnosis. Further direction, integration with microfluidic device and electrochemical readout to enable scaling in industrial use. From lab to field, next generation molecular diagnostic are improving how we can detect and how we can respond and even manage infectious threat and antimicrobial resistance. I would like to thank Dr. Ida Lab with my PI, Dr. Shibitoshi Ida, all the lab members, including for all this research, and would like to thank our funding resource. And we always welcome for a research partner and collaboration. And if you have any question or interest in any topic, you can reach out for Dr. Eda, or you can connect with me in LinkedIn or reach out for me via this email as well. For giving this, I would thank all you for attendance this seminar, and I will welcome for having any question. Thank you all.
Thank you, Dr. Sewid. Now I'd like to have some questions from the audience. Please speak up or leave your question in the chat. So let me ask one question, Dr. Sewid. What other One Health related issue this method can be applied?
So this method can be applied for detection of disease that affecting animal health, and also can be detecting any foodborne illnesses in affecting lotus or crops, and also it can be applied for any disease affecting human health. So it can be using this all disciplines with the same method.
Do you have specific pathogen in your mind for as a next target?
In my mind, in next target, I would like to test the integron. So like just like a mobile piece that can keep to carrying different antimicrobial gene resistant cassette and can be jumping from different species of bacteria or the same species as well and causing antimicrobial resistance. So this can be very good target to go ahead and test it.
Thank you. Any question? All right. Thank you very much, Dr. Sewid.
Thank you, Dr. Ida, for hosting, and thank you for giving me this opportunity and support me in the lab as well.
So I think we are going to finish early then. Thank you everyone for coming.
Thank you.
I saw Aga was there.